Funding for this program is provided by Annenberg CPB to advance excellent teaching.
I sent a message to the Congress, the first of this year, which provided for a national commitment for the conquest of cancer.
Much has changed since Richard Nixon declared cancer in 1971.
We've had a complete change in our understanding of what our basic makeup is all about.
We now understand the agents that are inducing normal human cells to grow like malignant cancer cells.
What have researchers discovered about our body's ability to protect us against cancer? How does a cancer cell live forever? And how has this research led us to a new understanding of the biology of the cell? It all starts with one renegade cell. Driven by accumulated gene mutations, the cell begins dividing out of control, creating millions of copies of itself, forming a cancerous tumor. About 40% of the people in the United States will develop some form of cancer within their lifetime. Half will be cured, and the other half will die from the disease. But since the 1970s, our understanding of cancer has radically changed. We now know that the development of cancer is a multi-hit process, requiring an accumulation of several gene mutations, usually over many years. We've also learned that cancer is seldom an entirely inherited disease. Though inheritance may play a role, most cancers result from a series of environmental insults. With this new understanding of the biology of cancer comes new possibilities for treatment. Judy is in her 4th year of treatment for CML, chronic myeloid leukemia, a rare form of cancer. Her treatment is Gleevec, 3 pills twice a day.
Gleevec works by targeting the specific abnormality that drives the growth of one particular type of leukemia. Gleevec binds to that abnormality and completely shuts it down, so it can no longer trigger cell growth. That's very different than chemotherapy, which just non-specifically kills dividing cells.
Hi, how are you?
Brian Drucker is the lead investigator in the Gleevec study. Drucker's studies show that because Gleevec targets specific cells, it's more effective and has fewer side effects than traditional chemotherapy.
Let's have you take some nice deep breaths through your mouth, please. And once more. For patients who are diagnosed relatively early, 96%, almost all of them, have their blood counts return to normal, and typically within 4 to 6 weeks. In almost 3/4 of those patients, when you look in their bone marrows, we can no longer detect any leukemia cells unless we use extremely sensitive PCR techniques. As you know, your bone marrow looks perfectly clean.
Yes,
so
it's nice to be able to say remission.
Yes. That's right.
I mean, I didn't think I was going to say that.
While Gleevec is still a rare exception, its development demonstrates a new understanding of the diseases we call cancer.
And thank you.
In the early days of cancer research, many scientists believed that all cancers came from viral infections. With the advent of molecular biology in the 1970s, researchers Mike Bishop and Harold Varmus made a breakthrough while studying cancer in chickens. Bishop and Varmus were able to show that cancer doesn't always start with the virus, but it does always start with the gene. But it was researcher Robert Weinberg who was first able to test this theory in human cells. Weinberg took DNA from a cancerous bladder cell and transferred this DNA into a non-cancerous cell. Soon, the non-cancerous cell began to divide in a malignant fashion.
Until that point in time, it was only a speculation that gene mutation was causative for human cancer. The discovery of a gene which was not only mutated, but carried the power of converting a cell into a cancer cell, provided the conclusive proof that the origins of cancer could be traced directly to mutated cellular genes.
The gene Weinberg discovered was called RAS. RAS is what is known as a proto-oncogene. Proto-oncogenes are genes that help tell a cell when to grow and divide. But when mutated, proto-oncogenes can transform into oncogenes, genes which can lead to uncontrolled cell division.
The proto-oncogenes are present in our genomes and in the genomes of all of our ancestors for the last 500 million years. They are there because they play essential roles in controlling normal cell proliferation. Without the proto-oncogenes, embryos wouldn't be able to develop, and adult tissues would not be able to be maintained. However, the price of carrying these proto-oncogenes with us in our genomes is that occasionally they become damaged and mutated and convert into oncogenes and thus become converted into agents for causing cancer.
Proto-oncogenes are like the gas pedal in a car, providing instructions for cell growth. When they mutate into oncogenes, it's as if the gas pedal is stuck in the down position. The cells are given the instruction to grow without stopping. Weinberg's study of the RAS oncogene revealed how the gene differs from its healthy proto-oncogene counterpart.
The difference between the normal human gene and the cancer-causing bladder carcinoma gene was very subtle. Out of 6 kilobases, 6,000 bases of DNA sequence, there was only a single base, a single nucleotide that was altered. And that single nucleotide alteration, a so-called point mutation, was sufficient to convert a normal cellular proto-oncogene into a virulent oncogene.
Point mutation, the change in a single nucleotide, is just one way proto-oncogenes can become oncogenes. Some cells become cancerous because proto-oncogenes are amplified, a process that greatly multiplies the number of growth-promoting genes, pushing the cell to uncontrolled growth. Other cells become cancerous because the proto-oncogenes are rearranged in a process known as chromosomal translocation. In this process, a gene from one chromosome is stuck together with a promoter region from another chromosome. The resulting fused gene can send a constant stream of growth signals. The RAS protein, the product of the RAS gene, is what is known as a G protein. It's a key component in the cell cycle as part of what is known as a signal transduction pathway.
It's a long, complex bucket brigade, and one of the partners in that bucket brigade is the protein called RAS. RAS receives signals from upstream in the bucket brigade, and in response to those signals, it passes them on further down.
Cell growth is activated when the cell receives a signal from phosphorylated. This begins a chain of reactions that in turn activates the RAS protein. The signal transduction pathway continues in a multi-step sequential process. Like a row of falling dominoes, each protein interaction triggers a new set of proteins, which triggers an even greater number of proteins. The final proteins activated in this chain reaction are transcription factors which enter the cell nucleus, generating a response, the transcription of mRNA. This mRNA is translated into a protein that triggers cell growth.
What we found in the early 1980s was that the protein made by the RAS oncogene is kicked on into the on position like its normal counterpart and sends out all of these growth stimulatory signals. However, the mechanism by which the RAS protein shuts itself off and puts itself in the down position is defective.
Without a functioning off switch, RAS continues to flood the cell with growth stimulatory signals, even without any external stimulus. Normally, these growth signals last only a few seconds, but when RAS is mutated, the signals can continue for hours or even days.
And therefore, a cell carrying a RAS oncogene and making a RAS oncoprotein is pushed to grow inexorably, unrelentingly, instead of being exposed to brief bursts of growth stimulatory signals. And that is one of the prime attributes of cancer cells, this unrelenting, inexorable growth.
Discovering and understanding oncogenes was a major step in understanding cancer, but it only explained one part of the process. In fact, it usually takes several different genetic mutations over many years for cancer to develop. Most of the mutations we accumulate are from the way we live our lives, smoking, the foods we eat, exposure to chemicals. But some mutations occur just from being alive. Each time our cells make copies of themselves, there's a small risk that a spontaneous mutation can occur. Imagine the scribes who used to copy huge volumes of text by hand. Even with all their careful attention to detail, with such a huge number of letters to copy, errors were bound to occur. But despite all the gene mutations we encounter in our lives, most of these mutations won't lead to cancer. That's because we have systems built in to make sure mutated cells either stop reproducing, fix themselves, or self-destruct.
The dark circles, they're the breast cancer patients. Which of them?
Mary Claire King is a researcher at the University of Washington in Seattle. Back in the early 1990s, King's work was crucial in discovering BRCA1 and BRCA2, key genes involved in breast and ovarian cancer. Today, King continues her groundbreaking work, further investigating these key tumor suppressor genes.
A tumor suppressor gene is a gene which can have any of a variety of functions that help a cell stay normally on a healthy course of division and development. These functions can be the repair of damaged DNA, the maintenance of genomic integrity. The reason that in the context of cancer, we call them tumor suppressor genes, is that if those functions are wiped out, now the cell no longer maintains the normal function that it used to have. So that cell type now goes on to be primed to develop a cancer.
While oncogenes are like the gas pedal in a car stuck in the down position, tumor suppressor genes are like the brake pedal. When tumor suppressor genes are mutated, it's as if the brake pedal is stuck in the up position, allowing uncontrolled cell division.
I've always felt a little queasy about the idea of calling these tumor suppressor genes because it suggests that the gene evolved in order to suppress tumors, but these genes didn't evolve in order to suppress tumors. These genes evolved to take care of the normal, healthy life of cells.
One of the most important tumor suppressor genes is P53, called the guardian angel of the cell. The mutated version of this gene is implicated in over half of all cancers.
P53 is critical because it protects the cell from a variety of other kinds of mutations that can occur. Consequently, P53 mutations are common to, that is, are shared by, a wide variety of cancer types, unlike many of the tumor suppressor genes in which one finds altered only in colon cancer or only in breast cancer or only in endometrial and colon cancer. P53 is altered in a wide variety of tumor types.
Damage to a cell's DNA leads to an increase in the amount of functional P53 protein present in the cell. The P53 protein is a transcription factor that works in several ways to ensure that damaged DNA isn't replicated. In one process, P53 activates transcription of a gene which codes for a protein that can stop the cell cycle. In another process, P53 activates transcriptions of genes that repair DNA. And in a third process, P53 activates transcription of genes that induce apoptosis, programmed cell death.
Once P53 is inactivated, then this entire alarm system is deactivated. And as a consequence, now cancer cells can proliferate unrelentingly without having to worry about interference from P53.
Because tumor suppressor genes provide this important cell protection, the path to cancer often begins with mutations to the tumor suppressor genes.
Early mutations in cancer are very often in exactly those genes that preclude other mutations from happening, or other mutations from taking hold when they occur. So mutations in genes that are critical to DNA repair are often the first step in what then becomes a series of mutations that leads to cancer.
Research has shown that while an oncogene can contribute to cancer with mutation to just one of its alleles, tumor suppressor genes must have both copies mutated before a cell can turn cancerous. However, King's research on the breast cancer genes BRCA1 and 2 has shown that a single mutated tumor suppressor gene carries a high cancer risk.
So if I were a woman who had inherited a BRCA1 mutation, either from my mother or from my father, I'd be just fine. But all of my cells would have only one functional copy of BRCA1 left. And the danger, therefore, is that any time a cell loses the one remaining surviving normal copy of BRCA1, that cell no longer maintains its genomic integrity. And that cell is now predisposed to go on and develop into a tumor.
Tumor suppressor genes and proto-oncogenes both work to ensure regular cell growth and division. But how are these complex processes orchestrated to work together? The answer is the mechanism known as the cell cycle. In its path towards growth and division, a normal cell proceeds through the 4 stages of this cycle: G1, synthesis, G2, and mitosis. Gaining greater understanding of how this cycle works has been an important part of understanding cancer. Leland Hartwell is the director of the Fred Hutchinson Cancer Center in Seattle, Washington. Hartwell won the Nobel Prize in 2000 for his work using the model organism yeast to study the workings of the cell cycle. Because of the underlying similarities in cell biology between all eukaryotes, Hartwell's work with yeast led to a more precise picture of the cell cycle in humans.
The basic orchestration of the cell cycle really requires solving sort of 2 kind of fundamental problems. One is that you have to establish order so that certain things occur before other things. DNA replicates before it segregates. And the second thing you have to do is coordinate the activities of very large numbers of proteins. DNA replication involves, 1,000 proteins probably, and chromosome segregation probably involves 1,000 proteins. So how do you get all of those to function together at the right time?
The answer seems to be in the cell's use of enzymes called kinases, whose action is enhanced by proteins called cyclins. To activate the kinases, the cyclins bind to them, forming a partnership that in turn phosphorylates other proteins specifically involved in DNA replication. These activated proteins move the cell cycle into its next stage. The cell cycle can't move forward until enough cyclins have built up in the cell. Once DNA replication takes place, the cyclins are degraded so DNA can't replicate again. An additional mechanism also helps ensure normal functioning of the cell cycle. At 3 different places in the cycle, the process stops at checkpoints to make sure the DNA isn't damaged or to make sure that the cell is vigorous enough to reproduce. If any part of the process isn't functioning properly, the cell cycle is arrested and the cell stops replicating. In this way, the process prevents the duplication of defective cells.
So that explains why things are ordered. So it's a very, very beautiful and simple story, undoubtedly too simple, but you can achieve some rather fundamental insight into the problem of coordinating many different things and then ordering different events with a very simple mechanism.
Just like the timer on a washing machine, the cycle proceeds on its own, driven by an internal clock. But in addition to the steady forward movement of the timer's clock, the cycle relies on other internal signals. The timer won't move to the next phase until the tub is filled with water. And it's dependent on external factors. The tub can only be filled if the external water controls are turned on and functioning properly. If any part of the process malfunctions, the cycle will stop. Hartwell's discovery of the cell cycle's checkpoints came about through the recognition that yeast cells stop replication when their DNA is damaged.
The cell is aware of the fact that its DNA has been damaged. It controls its cell division to wait and repair. And so we began investigating that phenomenon and discovered this control mechanism that recognizes damage and then controls division in response to it.
Hartwell's insights into the cell cycle have helped researchers understand the well-orchestrated growth of healthy cells and brought about new understanding of how damaged genes can lead to uncontrolled growth in cancer cells. But there's another crucial factor in the life of the cell that has only recently been examined. At the University of California in San Francisco, researcher Elizabeth Blackburn is studying a key factor in cancer's immortality. A tiny piece of DNA located on the tips of our chromosomes, called the telomere.
Chromosomes are linear. There are two ends, and it turns out that a DNA end is a very vulnerable spot for a DNA molecule. And so what the chromosome, what the cell has to do is protect that end. So that's called the telomere. It's a little protective cap, and people liken it to the little plastic tip that protects your end of your shoelace from fraying.
During cell division, the tips of chromosomes are not completely copied. Each time a cell replicates, the telomere becomes a little shorter. After a number of cycles of replication, the telomere has degraded so far that the cell can no longer replicate. But some cells, like germ cells, immune system cells, and stem cells, must keep replicating endlessly. In Blackburn's studies, she saw that in these cells, something was compensating for telomere loss.
And that something was the enzyme telomerase, which we discovered back in the 1980s. And telomerase has a special way in which it adds back extra little modules of DNA to the ends of chromosomes and keeps them from getting shorter and shorter and shorter. All the time you're growing up from a fertilized egg into a baby, telomerase is active a lot of the time. Your cells are doing lots of multiplication. And then when you turn into an adult, telomerase is turned down or maybe almost completely off in many, many cells.
But Blackburn's studies show that in many cancer cells, telomerase is reactivated.
The more advanced the cancer is, the more likely it is to have made a mistake in its regulation of telomerase, and now telomerase is just turned right on. It's like the rheostat got turned full on, and it's making a ton of telomerase. And a remarkably high percentage of tumors that people have looked at have activated telomerase. Numbers are put at, 90% plus of tumors.
Many researchers believe telomerase may provide one of the best targets for future cancer treatment.
So when you think about cancer, the big problem of cancer is it's part of us, right? So the trick with cancer is to try to tease out what makes the cancer cell different from the good normal cells. So I think the hope is to use the fact that so many cancer cells do turn their telomerase on full blast and have become reliant on that, and that is a kind of vulnerability that we think that they may have.
The presence of oncogenes, mutated tumor suppressor genes, and the reactivation of telomerase all appear to be necessary conditions for a healthy human cell to transform into a cancer cell. But researchers have identified another crucial process that seems to be a requirement for a cancerous tumor to survive.
The proliferation of a cancer cell in the presence of oncogenes and inactivated tumor suppressor genes, and even the telomerase enzyme, will go on for a little while, but after a while, a very small clump of cells, let's say 0.2 millimeters, will stop proliferating simply because the cells in the tumor mass don't have proper access to the circulation. In the absence of that access, the cells in the center of the tumor mass, small as it may be, cannot get oxygen, they cannot get nutrients from the blood, and they cannot eliminate all kinds of metabolic wastes, including carbon dioxide. The only way they can break out of this predicament is if they somehow acquire direct access to capillaries, to arteries and veins, which now supply them with an abundant amount of glucose and oxygen and other nutrients that will suffice for their further replication.
This process is called angiogenesis. Tumor cells send molecular signals to surrounding normal host tissue, encouraging the proliferation of new blood vessels. These new vessels supply the tumor with the nutrients and oxygen required for growth. Researchers are not clear what mechanism the cancer cells use to initiate new blood vessels, but many believe this is a rich area for new approaches to cancer treatment. Oncogenes, tumor suppressor genes, telomerase, angiogenesis. Each of these can represent a step that moves us closer to cancer. But new understanding of these areas also moves us closer to more effective cancer treatments.
The doctor said there was no time left and I'm still here.
Well, you're doing great. We'll get your blood counts regularly as usual.
4 years after starting Gleevec, Judy's cancer shows no signs of returning. And though this drug remains the exception for now, it's clear that our war on cancer is showing signs of success.
We have now this enormous backlog of information that we've accumulated for the last quarter century, and now we can begin to take advantage of it, together with new and modern ways of designing drugs, to really create from nothing very specific and targeted drugs.
When you're in the trenches at the bench trying to do experiments and frustrated, you feel like it's going so incredibly slowly. But then when you aggregate everybody's experiments and all of the insights over decades, you see that we actually are moving pretty fast.
I'm extremely hopeful. Gleevec has really validated decades of cancer research, and to me, it's no longer a matter of can we target cancer cells? We know we can. It's just a matter of when will we get the job done.
Decades of cancer research have shown us how we can prevent some cancers too. Over half of the cancers in this country could be prevented by stopping smoking, improving our diet, and cleaning up known environmental hazards. As cancer research continues, biologists are gaining a more detailed picture of how a healthy cell functions and moving closer to understanding how the diseases of cancer begin with just one renegade cell.
Funding for this program is provided by Annenberg CPB to advance excellent teaching. For information about this and other Annenberg CPB programs, call 1-800-LEARNER and visit us at www.learner.org.